Sorry, I was having a little trouble with switching. Thank you very much. My name is Debbie Benson Kennedy. I am an employee of CSL Behring. CSL Behring appreciates ICER's assessment of Etrenada oncogene desiparbovec, or for brevity, Etrenada, in recognizing its value as an innovative gene therapy for people with hemophilia B. ICER's clinical evaluation has concluded there is an incremental to substantial health benefit of Etrenada with moderate certainty in comparison to factor IX prophylaxis. Further, ICER's economic evaluation of Etrenada compared to factor IX prophylaxis revealed Etrenada was dominant treatment with lower cost and higher qualities at the model placeholder price of $4 million. ICER consistently found cost effectiveness up to a cost of $9.97 million for Etrenada in the base case analysis, highlighting the substantial economic value that Etrenada can bring to the health system in addition to its clinical benefit. We appreciate this opportunity to comment on ICER's evaluation. In this ICER assessment, Etrenada demonstrated its transformational impact for both people with hemophilia B and the health system. From the patient's perspective, ICER acknowledged that treatment with Etrenada resulted in a significant reduction in annualized bleeding rates, ranging between 64% for all bleeds to 80% for treated joint bleeds, as reported from the phase 3 HOPE trial, in addition to alleviating the pain and minimizing potential disability from bleeding events. Further, the reduction in burden of therapy from weekly or more frequent intravenous factor IX injection was highlighted as an important benefit, with 96% of participants in the HOPE trial discontinuing factor IX prophylaxis. Of note, 100% of the 52 patients with successful transduction were free from continuous prophylaxis. CSL Behring appreciates that ICER recognized the clinical benefit and cost effectiveness of Etrenada compared with factor IX prophylaxis. We do, however, have a few comments we would like you to consider. From the economic and health system perspective, CSL Behring is in alignment with ICER in regard to the key components in the economic evaluation of Etrenada and appreciate the demonstration of cost effectiveness in various scenario analysis. However, while we respect ICER's approach on single and short-term therapies resulting in a large cost offset, our perspective is that using an arbitrary number of $150,000 as a cost offset cap to derive the health benefit price benchmarks for Etrenada does not accurately reflect the value of the treatment options for hemophilia B. People with hemophilia B who would be eligible for gene therapy have a severe clinical phenotype. While the current standard of care for these patients, factor IX prophylaxis, can cost between $565,000 and $735,000 annually, it is a life-saving treatment that has lengthened life expectancy for people with severe hemophilia by fivefold. In absence of alternative treatment options, the assumption of one quality gain is inappropriate, given the drastic reduction in lifespan that would be expected if a person with hemophilia B were to discontinue factor IX prophylaxis. It appears in the process of highlighting the cost offset cap analysis that an edit was made renaming the base case to full cost offset. ICER has used base case consistently throughout its history, and without supplying context, this term full cost offset is highly likely to be misinterpreted. We politely request ICER to revert the wording to base case analysis for consistency with previous SST assessments. We would also request the order of analysis in the results table be consistent with prior reporting. Finally, in ICER's budget impact analysis, we are in agreement with the assumption relating to gene therapy eligible population. However, we believe the assumption of annual uptake of 20% of eligible patients dosed with Etrenada is unrealistic. This logic would assume also their patients receive Etrenada within 5 years. These assumptions lead to an overestimation of budget impact of Etrenada. Furthermore, we'd like to point out that a 5-year time horizon substantially underestimates the value of Etrenada. By year 6, all costs from Etrenada would be offset. In subsequent years, Etrenada would be highly cost-saving to the health system. We feel that adding comments in the report regarding the longer-term cost savings potential for Etrenada is justified. CSL Behring is proud to be serving people with hemophilia B with a novel therapy with greater clinical and economic value than the current standard of care. We commend ICER for a thorough and reasonable assessment, although at the same time ask that our comments be considered in the final version of the report. In closing, we thank the hemophilia B community for their continued collaboration and support. We will continue to work with stakeholders to ensure that both people with hemophilia B and the health system can benefit from Etrenada. Thank you very much.
Thank you, Dr. Benson Kennedy. And we have time for a couple of questions, if there are any from ICER staff or CTAF members.
So I just wanted to respond for ICER on that. So, had we not assumed any cap on cost offsets, you could end up with a price for etranadez of close to $10 million. And that doesn't seem particularly helpful from a policy standpoint. The $150,000 a year cap is assuming that cost-effective therapy would be about $150,000 a year at maximum. And interestingly, that's not very different from what's paid in many countries that aren't the United States for factor. So, when you're talking about life-saving therapy that could only be had at $600,000 or $700,000 a year, that's not true if you're living almost any place else. So when you're looking at what a fair price is, we thought that it would be helpful to people to see what a fair price would look like with that $150,000 cap. I also just wanted to very briefly mention on the budget impact. Our budget impact analyses are not an attempt to predict what the actual budget impact would be. They're an attempt to say what the budget impact would be depending upon what you assume the uptake is, to give a range of different uptakes up to 100% uptake. We in no way think that 100% of eligible patients will have uptake over 5 years. So just to understand as you look at that, and it is correct that over time, while there may be a budget impact up front, a therapy like etranadez would be cost saving. Thanks.
Any other questions for Dr. Benson Kennedy? Okay, we will move to our second commenter, Dr. Wing-Yen Wang.
All right, good morning. I am Wing-Yen Wang, Group Vice President in Worldwide Research and Development at BioMarin. And on behalf of BioMarin, I appreciate the opportunity to provide comments on ICER's review. As this audience is aware, severe hemophilia A is associated with spontaneous bleeding and painful disabling arthropathies. Prophylaxis, as you've noted, with factor VIII or emicizumab requires routine administrations of 100 to 150 infusions or 12 to 52 injections per year, respectively, again costing well over $600,000 annually. In a large prospective non-interventional study, greater than 75% of participants reported bleeding episodes with a mean annualized bleeding rate or ABR of 5, despite over 90% adherence with prescribed factor VIII regimens. Suboptimal adherence persists in the real world, further compromising outcomes. GENERATE-1 resulted in over 80% reduction of ABRs requiring treatment and an 98% reduction in use of factor VIII, demonstrating that gene transfer with lactocogene roxaparvovec, or as you call it, Valrox, provides superior bleed protection relative to repeated exogenous factor VIII prophylaxis. It's worth noting that 74% of participants had zero treated bleeds and over 95% remain free from prophylaxis during the 2 to 3 years of follow-up. Participants demonstrated broad and consistent improvements in health-related quality of life sustained for 2 years post-gene transfer. Measured by the hemophilia-specific quality of life questionnaire for adults, clinically meaningful improvements were reported in the domains of role functioning, consequences of bleeding, worry, and treatment concern, as well as less classroom and work impairment, and the largest improvement in leisure activities and sports, suggesting increased physical activity and strength after gene transfer. Now, Valrox has demonstrated an acceptable, well-tolerated safety profile to date. Transient asymptomatic grade 1 to 3 transaminitis and transient infusion reactions were the primary side effects. Patients had no symptoms or sequelae suggestive of clinically significant hepatocellular injury or liver dysfunction. ICER had raised concern about the rate and prolonged use of corticosteroid treatment in the clinical trials. Corticosteroid initiation for GENERATE-1 was per the investigators' discretions, and we acknowledge a propensity for corticosteroid use. We expect that immunosuppression will be less intensive in the post-approval setting. And the European label reflects a more restrained use of corticosteroids. We will monitor the use of immunosuppressant regimens in the real world with comprehensive 15-year follow-ups, as well as a post-approval registry. While BioMarin appreciates ICER's recognition of the long-term value of Valrox, BioMarin respectfully disagrees with ICER's approach of applying the $150K per year cost-saving cap to set the price. Upon using traditional cost-effectiveness framework, ICER concluded that Valrox price at $2.5 million is projected to be associated with lower costs, lower bleeds, and higher quality-adjusted life years compared to emicizumab. The results are robust across all sensitivity analysis and in all simulations at all willingness to pay thresholds. Even when applying 50-50 shared saving scenario, Valrox remains a dominant treatment strategy. We do recognize the financial burden or medical debt of current hemophilia treatments and understand the motivation behind the shared savings scenarios. While ICER can apply shared savings framework in a scenario analysis as part of ICER's single and short-term therapy framework, ICER should not set price recommendations based on these arbitrary thresholds. The annual cap of $150,000 cost saving is far below the real-world cost of prophylaxis, which exceeds $600K per year in the United States. The outcomes-based warranty agreement that BioMarin plans to offer for Valrox is designed to address variability and durability concerns for payers and health systems. It also reflects BioMarin's belief in the clinical value of Valrox and our willingness to stand behind the value proposition and compensate payers and health systems in cases of suboptimal responses during the warranty period. Now, following the approval of valoctocogene roxaparvovec in Europe, a clinical trial participant shared his personal perspective. He stated, "The impact of gene therapy on my day-to-day has been a life change. It has been a big change. Try a little what I have never had. The peace of mind of living day to day without being afraid of everything. That's the 3 years I've had so far, a normal life." So thank you very much. We appreciate the engagement we've had so far with ICER and really look forward to continuing to work together and with the community. Thank you for your attention.
Thank you, Dr. Wong. We have time for 1 or 2 questions. Rita.
It's not a question, but more a comment. And the comment is just because Dr. Wong referred to comparative costs, but I would say that costs in the US health system are widely inflated and not really a fair comparison. You know, factor costs are obviously very high. And comparing to cost of manufacturing production, even including research, looking at costs in other countries, I think saying that you want to make a comparison based on a very inflated, dysfunctional model is not in public interest.
Bob.
Okay, you've got my attention with the outcomes warranty statement. I would appreciate it if you'd flesh that out just a little bit more and specifically comment on how that in any way directly affects the patients.
I believe we will have a larger discussion. This is a complex issue, of course, in terms of OBAs and warranties, and we'd love to share that information with you. So I think that it would be more appropriate for a larger discussion, a great dialogue within the discussion period. Happy to do that. Thank you.
Don't worry, Bob. We'll pick up on that for sure.
Okay, if we do not have any other questions at this point for Dr. Wong, I will turn it over to Dr. Richard Ko.
Can you hear me?
Yes.
Great. Just to disclose, I am an employee of Genentech, and we, on behalf of Genentech, thank ICER and the panel for their time and attention. So good morning. My name is Dr. Richard Ko, and I'm an executive medical director in U.S. Medical Affairs at Genentech. As a pediatric hematologist with over 15 years of experience in hemophilia, I'm truly inspired by the innovative treatments that are being developed for people with hemophilia A. At Genentech, our reason for engaging ICER during this assessment of the investigational medicine valoctocogene roxaparvovec, which I'll call Valrox from here on out, is to ensure that people with hemophilia A can access the treatment that is right for them. Our comments today will focus on the hemophilia A assessment since Valrox is being compared to emicizumab. We appreciate this opportunity to summarize the evidence that we think is pertinent to this assessment. Emicizumab is FDA approved for routine prophylaxis to prevent or reduce the frequency of bleeding episodes in adult and pediatric patients ages newborn and older with hemophilia A with or without factor VIII inhibitors. Over 15,000 people with hemophilia A worldwide have been treated with emicizumab. Results of the Haven clinical trial program have repeatedly demonstrated that prophylaxis with emicizumab has led to significant reductions in annual bleeding rates compared to on-demand and prophylactic factor VIII therapy. Additionally, the majority of people in the Haven trials experienced zero treated bleeds within the 6 months after starting emicizumab. More than 5 years after approval, several real-world studies associated with emicizumab with annual bleeding rates that are as good or better than trial outcomes. Real-world studies have also highlighted additional benefits associated with emicizumab, such as reductions in treatment burden. Cost-effectiveness analyses published separately by Genentech-affiliated researchers and ICER-affiliated researchers have shown that emicizumab is a cost-saving treatment when compared to factor VIII prophylaxis over the lifetimes of people with hemophilia A without inhibitors. Moving on to the current ICER assessment, we agree with the clinical assessment in which ICER determined that there is insufficient evidence to compare Valrox to emicizumab. As we have outlined in our written responses to the published drafts throughout this review, we do not agree with the conclusions of the economic assessment. The patient journey and clinical outcomes portrayed in the economic model are not consistent with what was reported in the GENERATE trial. Specifically, within 2 years after receiving Valrox in the GENERATE trial, approximately a quarter of patients had factor VIII levels in the moderate to severe range. However, the base case economic model assumed that there would only be a 2% treatment failure rate per year in the first 4 years. By not accounting for the heterogeneity in treatment responses seen in the GENERATE trial, the economic model overestimated benefits and underestimated costs for Valrox. Similarly, the base case economic model did not incorporate the severe adverse event rate that resulted in hospitalizations of some people who participated in the trial. This artificially lowered the overall costs associated with Valrox. The economic assessment also did not reflect the standard of care for initiating prophylaxis. Although there is no current consensus on when to reinitiate prophylaxis when the benefits of Valrox diminish over time, current clinical guidelines should be applied to this situation. Quoting the MESAC guidelines, prophylaxis should be individualized and sufficient to prevent all bleeds at all times. What ICER modeled would result in more bleeding events, as many as 7 each year, before a person would restart prophylaxis. This would not reflect an appropriate standard of care, especially for those who were previously doing well on prophylaxis. It is essential to build economic models that reflect the standard practice and relevant clinical guidelines. Lastly, instead of allowing for prophylaxis to be tailored to the individual as per the guidelines, the economic model only allowed patients reinitiating prophylaxis to take emicizumab, not factor VIII products. This would not reflect clinical practice since according to the Athens registry, almost half of patients on prophylaxis in the US are using factor VIII products. The various different treatment pathways that occur in the real world need to be reflected in economic assessments to best inform decision makers. In closing, treatment decisions and coverage determinations about Valrox could have long-lasting impacts on the lives of people with hemophilia A and the treatment options available to them. For example, patients who utilize Valrox may not be eligible for future AAV-based gene therapies. Therefore, it is critical that today's discussions provide decision makers with the information needed to ensure that decisions on future budgets and covered treatments can meet the needs of people with hemophilia A. Thank you for your time, and we appreciate the opportunity to comment.
Thank you, Dr. Ko. We have time for a few questions for Dr. Ko, if anyone has them.
I'll just answer also from ICER. So first, we agree with much of what Dr. Ko said there about the benefits of emicizumab and the uncertainties with gene therapy and with AAV therapies, potentially preventing future AAV therapies. We agree that those are concerns. On the economic side, the issue is that all the things you talked about there aren't going to change the answer of the economic analysis, which is in the full cost offset model, the gene therapy will be cost saving compared with emicizumab over a fairly short period of time because of the cost of emicizumab. If that cost came down, then maybe there are arguments to be had about edges of the model, but that isn't what's happening here. We're talking about a therapy that if it was priced at $2.5 million would be the same price as emicizumab over about 4 years or less. So that's really where the issue is. And whether you really think that our 2% per year failure, which we got from the manufacturer, is exactly right, or whether there should be different numbers, isn't going to change that very much. And ultimately, the comparison of going back to your product rather than factor VIII is because we've concluded previously that your product is better than factor VIII, it's dominant over it, and that's what we think the right path of care should be. And we think you all should be happy with that.
Thank you, David. Any other comments or questions for Dr. Ko? Okay, we have 2 more commenters. We have Will Hubbard and Nathan Schaefer. And reminder to please mention your disclosures, and we'll start with Will.
Thank you, Raina, and thank you, ICER, for having me today. As my conflicts slide shows, I work for the National Psoriasis Foundation, which does meet ICER's threshold as an organization that receives contributions from pharmaceutical companies. However, I am speaking today in my personal capacity as a person with hemophilia, and my views should not be taken to reflect that of the organization. So a lot of great points have been made already. I just want to briefly add my patient perspective on 3 decision-making factors at play in my decision, if you'll excuse the pun. And I am currently someone in the wait and see camp when it comes to gene therapy. I'm certainly heartened by some of the results, but there is more I would like to see, and I'll briefly explain my thinking here. So the first point I want to make is about the importance of transparency and risk communication, not just in clinical trials, but throughout the process as these come to market. Some of my friends have taken gene therapies and participated in trials, and they have done a ton of due diligence in terms of researching the risks and potential benefits of all these therapies. And to their credit, the researchers and companies underwriting and leading these trials have also been transparent about both the risks, the benefits, and the uncertainties. And I'm encouraged by all of this. I would just add that this needs to be part of the process going forward, even after FDA approval for any of these therapies, right? Lots of these risks are relatively novel for people with hemophilia. And so there needs to be robust communication that is constantly updated, the materials used to convey these upsides, downsides, and risks to reflect new scientific evidence. These therapies are going to, people with hemophilia like to remain educated about the standard of care and the disease itself. Gene therapy is going to push the boundaries of scientific knowledge that patients are asked to bring to bear. And I think it's important that all the stakeholders in this mission are partners in helping patients get up to speed in that respect. The second point I want to make is that, as has been stressed throughout this, these are all long-term therapies, right? These gene therapies potentially permanent. And so when you're evaluating that as a patient, it's not something that you can think 2 years out, 4 years out. If there are long-term safety concerns, for instance, the risk of cancer, these are all things that we have to weigh. And if the therapy isn't a lifetime thing, if it is just a vacation from prophylaxis, for instance, that too is something that we need to know. And as has been demonstrated, there is a range of possible outcomes for people with hemophilia when it comes to the bleed rates and the factor rates they have in the long-term follow-up from these studies. So I say this to stress that there needs to be long-term surveillance and follow-up with patients who participate in gene therapy, both at the trial level and once it comes to market. This can't be a one and done thing. We need to continue following these patients and assessing their health outcomes, because otherwise we risk iatrogenic events and patterns falling through the cracks or not being picked up early enough. The final point I want to make is that, like emicizumab, I apologize, that's always a tongue twister for me. There are other non-factor replacement products that are in the pipeline that are coming to market. And I would just encourage this group. I mean, I'm sort of preaching to the choir here. ICER, more than anyone, is interested in the relative utility of different drugs relative to patient outcomes. But this is just something I think about a lot as a patient. There are going to be other options in this space coming out, and we can't be dogmatically focused on one thing or another. Gene therapy provides enormous potential, especially when it comes to a cure. Other products may have advantages, especially in parts of the world where the cost of factor, let alone gene therapy, is prohibitive. And so I would just say that hemophilia is a diverse disease with a number of underlying mechanisms and comorbidities and complications. So we can't be dogmatic about a one-size-fits-all approach. There will be nuance here and different patients who do better with different therapies. And that's something that also should be communicated clearly to patients throughout this process. That's the extent of my comment. I just wish to thank the committee for their time. I'll take any questions.
Thank you, Will. Questions?
I'm not sure if I can make a comment.
Please.
Okay, I fully agree with the, what the information that Mr. Hubert had provided. I just want to make an additional comment in regards to the safety and the long-term follow-up. Right now, there are definitely strategies that are being put in place coming from different organizations, including the World Federation of Hemophilia, to do long-term follow-ups, including registries that are very detailed, and every single patient that gets involved in gene therapy. It could either be from research or when any of these molecules do get approved. And there are also many registries that are starting in specific countries, including North America and Europe as well. So definitely there will be very close follow-up.
Bob.
I click that thing one way or the other. Well, I appreciate your comments. It's common for us and the other ICER panels to have patient communities just screaming for the newest and latest and greatest thing, and they're always better. So I appreciate your balance of actually trying to communicate that's not always true, and there are upsides and downsides to the whole thing. I'd frankly just like to hear you talk a little bit more about your awareness from the broader community of the feeling about these things and their sort of positioning. I understand that the hemophilia community has been sort of burned in the past by great promises and things. So if you just talk a little bit more, I'd appreciate it.
Yeah, certainly. So, as many people in this group know, and as was alluded with the Hep C comment earlier, hemophilia is ground zero for one of the largest iatrogenic catastrophes in the 20th century with contaminated factor products. When first hepatitis and then later HIV entered the blood supply, the hemophilia community was incredibly hard hit, with over half of all Americans with severe hemophilia infected with HIV through clotting factor, and over 10,000 across the country. And that certainly weighs on this, but I would also say there's tremendous optimism, right? We know that mistakes of that era have been grappled with and internalized, but it certainly colors the community's perception. The other thing I would say very briefly is that different community members come to this opportunity with different incentives and different sort of hopes and anxieties, maybe because of their history of care. So for instance, I was born after all this occurred. I was on a pup study for recombinant, which means that I've never been on human product since my birth. I've also had very well-controlled bleeds. I've been very fortunate in my standard of care with factor and emicizumab. So my joints are pretty healthy, and I don't feel a burning need to switch to a gene therapy. There are other people for whom that's not the case, who have inhibitors perhaps and aren't having their care effectively managed by current offerings, or do have severe joint damage, or even think that they can't stay on insurance long-term for one reason or another, have that anxiety, right, and would prefer something that doesn't require that continuous coverage. So all patients will have individualized concerns and come to this with different priors, and that really colors how they see this. It's not just the history, though that is a major part of it. It's also each member's individual disease.
Great. I think we have time for one more. So let's say Sanket.
First off, thank you so much for coming and sharing your experience. It's so invaluable and just so extremely helpful to us. I wanted to go back to the fact that, for example, you mentioned that you weren't involved in as a patient in any of the trials, but other friends, colleagues in the community were involved. I'm wondering if you might be able to speak to, I realize it may be hard to do, but to individuals who may have enrolled in the trial and perhaps why. And the reason I ask is because we don't have a randomized controlled trial. We have a pre-post analysis, and so the people who are actually enrolled in the trial may be different, for example, than those from the community, for example, may be looking for a solution and maybe sort of, for example, dissatisfied with current care. I wonder if you might be able to speak to that if you're able to, based on other colleagues in the community. Thank you so much.
Yeah, of course. And again, this is sort of idiosyncratic, so I hesitate to say that the observations I have are too, broad ranging. But for some people, they have a longer history of joint damage. Some of them, it's their individual assessment of the risks, benefits, and uncertainties, right? I personally don't have a background in science. Some of my friends do and are hardened by the prospects. Others do and still want to wait and see more. So there's I don't want to say that, a background in science leads to any specific opinion there. And then finally, some people just the long-term aggravation of constant infusions, right? I think that there's a lifestyle component to that as well. So those are a couple of reasons. And again, I don't want to speak on anyone's behalf because this isn't something I've discussed too much with people besides the scientific component, right? What do you think of the current research? I haven't gone too deep in the weeds of the stuff, whether it's lifestyle or whether it's community history. That hasn't been conversations I've had too much of, but those are just a few things off the top of my head.
I'm sorry, I'm going to have to hold on further questions so that we can finish. We have one final commenter, and any other questions I hope we can hold for the policy roundtable. Let me turn it over to Nathan Schaefer.
Great. Thank you so much. Thanks for the opportunity. My name is Nathan Schaefer. I'm the Vice President of Public Policy at the National Hemophilia Foundation, and I'm pleased to offer comments today on behalf of NHF as well as the Hemophilia Federation of America. First, as a reminder, we do not advocate on behalf of any given product. Since there is no one-size-fits-all approach that works for hemophilia, an individualized patient-centric approach to assessment and treatment is necessary. Second, thank you to ICER for incorporating many of our suggestions into your analysis, such as acknowledging important differences between hemophilia A and B and their therapies, recognizing health utility and patient important outcomes far beyond annual bleed rates, and considering patient and caregiver burden for current and novel treatments. Third, NHF and HFA remain very concerned about the economic burdens of all hemophilia treatments. We are on record stating that the current cost of hemophilia treatment is financially toxic for patients, their families, and the overall healthcare system. We want to underscore that finding gene therapy to be cost-effective does not mean that it is affordable, accessible, or optimal for every patient. We fear that high target prices will impede access to these potentially transformative therapies and caution against justifying U.S. prices based on a multiple of current treatment costs. Fourth, we are concerned about additional non-cost utilization management policies that will impede access to these therapies. For example, some payers have released policies that require patients with hemophilia A to fail on Hemlibra before accessing gene therapy. We also worry about barriers to the return to prophylaxis for people with hemophilia A, if and when this is necessary post-gene therapy. Medically appropriate treatments must be available to all patients without barriers due to inappropriate utilization management strategies. Fifth, we wish to emphasize that these novel therapies introduce complexities and unknowns that require ongoing patient monitoring and follow-up. In the US, this expertise exists exclusively within the national network of federally recognized hemophilia treatment centers. This is why NHF submitted a citizen petition to the FDA requiring that any approved hemophilia gene therapy product must have a REMS program, making gene therapy available only in the HTC network, along with mandating training for shared decision-making and long-term monitoring of potential adverse events. HFA, the European Hemophilia Consortium, and the World Federation of Hemophilia have also endorsed this request. I will close by quoting the first letter that we sent to ICER for this review. Hemophilia's complexity, the disease and treatment burdens associated with the disorder, the variations among patients, and the potential rewards and risk of any novel therapy demand an individualized, patient-centric approach to treatment. Patients and doctors should have the opportunity to select a treatment that meets patients' individual goals, physiology, life circumstances, and risk-benefit assessment. There will be patients who will adopt these potentially transformative novel therapies at their earliest opportunities, and other patients who, for equally important reasons, hold back. All must have access to the clinically appropriate therapies that best serve their needs and treatment goals. On behalf of NHF, HFA, and the people with blood and bleeding disorders that we serve, thank you for considering our comments.
Thank you so much. And we have time for a few questions, maybe 3 or 4 minutes left. Bob.
Okay, I hate to keep talking. I don't usually do that right now at these meetings, but I would challenge your use of the term patient-centric. If you're really advocating putting these gene therapies within the network, whatever network that may be. I understand that. I come from the cancer world. I understand the urge to do that, but part of the problem with this disease is people all across the country with not easy access to a network location. So I'd appreciate you trying to justify that being patient-centric to me, if you would.
I will say that not all patients have access to specialized providers all across the country. That's a challenge that we've encountered for many years at NHF and within our chapter network. So I acknowledge that not every patient has equal access to specialized care, is what I would say in response to your question.
Brian.
Thank you. I wanted to actually respond to the question that Sanket asked to Will earlier, because I am a person with severe hemophilia B, but I'm actually participating in a gene therapy clinical trial. It's over 2.5 years since I had gene therapy. And just in terms of my decision drivers for that, I mean, there are a lot of potential therapies for hemophilia. My treatment for factor IX deficiency prior to gene therapy was very good. I was on an extended half-life factor IX concentrate. I already had a very low ABR of 0 to 1, so the ABR was not the driver. And my drivers were, first of all, I followed the science very closely, and I was convinced by the science that the benefit risk ratio to me would be worthwhile. I wanted to, my ideals were, I wanted to stop the need for prophylaxis. I hoped for decreased chronic pain in my damaged joints. I wanted to spend less time on my own hemophilia and more time on everybody else's hemophilia as part of my job. I wanted to see if I could increase my activity levels. Also, given my age, I was 62 at the time. I didn't want to wait several years, perhaps, for licensed gene therapy and then go through the whole rigmarole around potential reimbursement. And I wanted to try life without severe hemophilia. Finally, I wanted to lead in Ireland because we've been talking about participating for a number of years. But I will say it was a very carefully considered decision. And I think for the hemophilia community, I think ICER and everybody else needs to be aware that we are going to be looking at very detailed education programs. This is an irreversible one-off decision. This is not going to be a rush decision. And I think all the patient organizations will be very committed to having a full series of education programs in collaboration with clinicians to make sure that people, if it's an option for them, that they don't rush into a decision that is very, very carefully considered and all the ramifications are looked at.
We are right about at time, but I would like us to be able to hear Mark and Felicia's questions if we can be brief. Mark?
Yes, this was really just a follow-up to Nathan's response on the patient centricity. One of the roles of patient advocates, patient organizations, is to look out for the best interest of patients, which is patient-centric. Given the significant burdens that come post-gene therapy, the once-in-a-lifetime opportunity, which is different than other therapies that may have been considered by ICER to date. We think it is patient-centric to require or to strongly encourage that all gene therapies for hemophilia begin in the treatment network. That is the network that we have today. Well over 80% of the US population is seen annually in a network, and they can work through a hub and spoke approach, and there's a well-detailed plan. So I think this is very consistent with patient centricity to keep it aligned around the education, the safety, as well as the expertise that's required to maximize the benefit given the once-in-a-lifetime opportunity.
Okay, Felicia, thanks.
In thinking about patient-centric decision-making, I can't help but wonder about the influence of insurance. How much is that a factor in patient decisions? Personal experience.
I think that is a very loaded question, and one that certainly speaks to what our reality is going to be like for the next couple of years. Because these therapies that are coming to market that clearly can be transformative for patients are very hopeful. How we're actually going to get them paid for and how we're going to educate policymakers and payers about it. We've got our work cut out for us, but we're up for the challenge.
Thank you. I'm going to turn it back over to Dr. Pearson.
Great. Thank you so much for those public comments from all perspectives. And I know we will touch on many of the issues raised when we return to the policy roundtable after the voting and the discussion that resumes immediately after lunch. So my job is to be quick here. We do have this odd timing, but we want to make sure we have time for the discussion to come. So if we can reconvene at 11:50 Pacific, that's 2:50 p.m. Eastern, obviously. So right now, please go grab your lunch and we'll restart right at that time, 11:50 p.m. PST. Thank you.